Okay. Um. So, hi, and this is the end of the May, and we're going to um have a long holiday, and it's a Memorial Day on next Monday. So finally, I got some time to continue my optogenetics series. So today I'm going to move on to the part 3, and it's about optogenetics in biological system. And today we're going to talking about how to visualize and manipulate of intracellular signals. So firstly is the outline. So we're talking about how to probing intracellular signal, and majorly on this part, we're going to go through to review literature and talking about other optogenetic tool for manipulate the signals. And we're in this lecture, we're going to revise and several chapter we're talking about before, like chapter 11, and it's talking about including and some of the contents of the chapter 11. So we all know that um, in our optogenetics toolbox is already have a lot of like candidates like rhodopsins, that's the earliest one. Then cryptochrome and that have a LOV domain, a light of voltage, an LOV domain, and also phytochrome and also bacterial chromes, and this all goes through in previous lectures. And today, firstly, we're talking about how use the light to investigate or to observe the kinetic signal pathway. That's is the ERK and ERK signal pathway. ERK is the downstream or the target of the MEK. And MEK and MEKK is considered like a ERK's kinase, upstream kinase, and it's a response to tyrosine kinase. With the cell stress, and it will activate this signal pathway and cause some more new gene expressions as a cellular response. So ERK and MERK signal pathway may also cause like proliferation of the cells or migration of the cells, and this all can be controlled by light. So here first, they use some molecular tools, and without light, without the ERK signal, and the cell response will be quiescent. And with the pulse stimulation of the ERK signals, it will cause the cell's proliferation. But if it continues the ERK, then this cells becomes like the being habit and become the transient again and stay in its cell conditions and without proliferation. In another cell function moves is that it may cause the cell migration. So for all of these cells, the EIK activation waves is first from here, then like for example, these two cells expression EIK first is activate first, then go to these two and then these two. And this is the ERK signal activation wave. But there's attraction force these cells migration from these directions. For example, there's a signal that activate the ERK here. And firstly, of course, these cells will activate the ERK and start migration. And then next, these cells. So the ERK's activation waves, like a prevail step by step from this direction, and the cell migration is from this direction. So this is like an example that this phenomenon can be observed with optogenetics tools. Later on, these chapters introduce the three very important database. The first one is the FP base, that's the fluorescent protein database, and the link is here. And click the link, and there's including a lot of like fluorescent protein like GFPs, and including a lot of informations like excitation, the curve, and emission, the curve, and a lot of qualities like a pKa, lifetime, temperatures, and the literatures is all included in this database, and this is very useful. And also there's other two, and there's also the other two database, and another one is the protein database. Okay, here is show that it's a FP base and have a lot of informations. And another one is opto base, and the link is here, and it includes every. So we can go on to see that at the most the response, the light wavelengths, and also labeled with how they respond, say LOV, and the origin like hypochromophone or phytochromophones, and all it's including here. And we can already find that those ones that were very familiar to us. Oh my God, it seems not now. It's like a CR2 and that's LV domains and binding with FAM or FAD. And all this is LOV domains. So, and so third database is a signal network. And I'm going to general review this article because it including several fluorescent biosensor to investigate different cellular activities. So this is summarized here that there's many tools that can using the fluorescent light to indicate like the calcium, like cyclic AMP, and some kinase function, and even more like some apoptosis or mitosis or meiosis, like the cell circles. They can all be observed with fluorescent light. So the GNSA is a fluorescent protein-based biosensor. And for example, here this shows that a membrane bound PI3K and 118 ATP, and these signals will go to the cell membranes. This green fluorescent. So it can indicate that this signal is being activated, and the fluorescent will coming out from the cytosol to the plasma membrane. This is described like a translocation based fluorescent biosensor. So like AKT JNKP38, also ERK, we already mentioned that after with the kinase, and the protein will be phosphorylated, and you will prefer to be in a cytosol. But if this phosphate is being removed, and you prefer to be located in the nucleus. So also this like the chymodulin sensing is a chymodulin REMEPAC PDE5 alpha, and with the reporters that it looks like this. So after adding the calcium, and this I think this circular pre-mutant GFP that after binding the calcium with the CAM, they will cause this fluorescent protein to be an intact one, and it can emit fluorescent light. So also this one can respond to CMP. And this group is for like response to cellular analytic and membrane potential. So this was including like voltage. This is a pre-mutant second GMP, and this GMP is in the extracellular side. So when the membrane potential change and the voltage sensor domains structures conformation change, and it will cause is the GFP's structure have a change, and it can respond to this fluorescent light like a decrease will indicate that there's a depolarization happens in these cells. And it can also be response to the calcium as another biosensor. So this is considered as the single fluorescent biosensor, and it can to indicate the membrane potential. And also the flat-based reporter for ions and membrane potential. And this is being reported in this literature. And generally said is CFP and YFP. And without the signal, then it's released, and the signal will be blue light. And after, like the conformation change and binding with the calcium, and this to approach the relative distance decreased, and also these two proteins are aligned together. The angle is also important for them to have this energy transfer from the CFP to YFP. So after there's a conformation change, and then this signal will be a yellow light. And this can respond to the gene as it's like how to cause the conformation change, and based on intracellular biosensor. Also, okay, this one is also the CIVSP and the KB2.1 best. This discussed in chapter 12, and it's based on frets. Member potential, and this is the flip is summarized in few minutes ago in this lecture, and that's it has a pH that can bind into a membrane bonding Pi3K, and cause there's some fluorescent light wavelength change. So all these seem to like response to the calcium, zinc, or like membrane potential here. And this is a hormone receptor. So after binding with its ligand PTH, and so this receptor is a ligand gated receptor, and it will cause the signals change. Also, there are other studies that's based on beta-2 adrenergic receptors is also in under this concept that after binding the ligand, the conformation change will cause the light wavelength change due to the frets. So this is the binding pocket is a rest. So when change the GDP with the GTP, it will also cause some change. And this is the rest and the IF RBD binding domains. And this respond to like a pKa activity, pKc activities. This all there's a phosphorylation site within this linker. So after like have some for phosphorylations, and it will cause the binding or dissociation of this component, and it will change. So the kinase will cause the blue light signal to change to the yellow light signal. So this can respond. So this like a kind of like a substrate, and it will respond to these kinase activities. So also histone transferase at after like adding adding an AD, and it will cause the conformation change, and this can respond to signal like the gene expression and transcription. And this is the DDFP. DDFP is response. So this is intercellular. So in inter like, so that means that there's a two parts of the component of the frets. But generally, most of the previous is they're only like linked together, and this light transfer is intracellular. But this DDFP like a double proteins that after binding together, and it will cause these two. Previously, they were not colocalizing was not fusing, but after adding the calcium, and due to the binding of this CAM and M13, will cause these two approach together and then cause have a fluorescent light emission. Also, this one that's without binding, that's then these two parts were far away, but after binding with calcium and then CM combining with M13 and this approach. So this DDFP and fluorescent best biosensor. So this is like amplifier, use luciferase, and it will use a substrate. So this process is require energy. So it require the cyclone AMP layer, and to amplify the light intensity that produced by the firefly's luciferins. So this part I think is pretty cool. It's a coupled reporter system that can analyze the mitosis. And this is Zhang work. And this chapter is also written by Zhang lab. And so Jenny said that on the G2 status that this MKO2, because it has a red fluorescent light, and it's been degraded in ubiquin dependent process, and so there's only the green fluorescent preserved. So in the nucleus, it's a green. So like in the nucleus, it's a green. But after turning to the M phase, both of the probes are degraded, and then there's no light. So it's dark. During the G1, the MAG will be degraded, and only MK2 will preserved. So the nucleus will show an orange color. And when the transfer from the G1 to S1 conditions, so both red and the green colors preserved, and these cells will be shown like in the yellow color. So this is pretty cool. That just show that different probes, because they preserve, because they will be degraded in the different stage of the mitosis, and so with both proteins preserved in the cells and with the color, and we can tell at what stage of the cells. I think this is pretty cool. And in 8 years later, they add more colors, one more colors, and this is the toolkit, so you can like separate those two periods of the mitosis even more clear. Like in S conditions, like first it have a three color like okay, when you see that is the toolkit is status is the toolkit preserved, and also both red and green have and yellow have orange color, and later the greens are totally gone. It's almost gone, and also the greens only happens here. And at this stage, is has the MK2, and at this stage, there will be two crystal tubes. So you can further separate these different conditions that can and tell as a difference between the S status and the G2 status. So there's even more, because we know that for the fluorescent, we can use a different filter to only see a single color. So we can like block out other wavelengths, and we can only see the blue one, and then we'll know that, oh, okay, this is in the S stage. And if we add in more filter, like to see a merge image, and we see if there's a green shows up, that means that at the late stage of the S. And if in the early stage, and we know that it's between the G1 and the S, and here there's orange and also red. So I think this is pretty cool. That can tell the cell cycle of the mitosis. So here, and this one, it can show that these biosensors also designed for like the synapse like tokens, the coupled reporter systems. It can tell that, for example, the neurotransmitter BDNF or SH is released. It will cause the target cells fluorescence change, and so I think that's a pretty cool. And also, this is a Caspase 3. So after shading with the light, then if there's also adding with the chromophore, and it will cause a fluorescent emission. So previous is a review at and all these contents are from these reviews. And later on the chapters, I will describe that the translocation-based biosensor, like first the ERK and MEK signal pathway. So after phosphorylation, the ERK by MEK, and the ERK will be translocated into the nucleus. And after the phosphorylation, and it will be nuclear export. And another kinase translocation reporter system, KTR system. In this system, they have a kinase docking domain and have a both signal, like NES and NLS. NES a nuclear export signal, and NLS a nuclear location signals. So with the dephosphorylations, a nucleus will be import. So within here, so only this green fluorescent will be light up. But after adding the phosphorylations, and so this export signals is being used. So this the whole protein was being located in the cytosol. So dephosphorylation go inside, phosphorylation go outside. And this translocation, phosphorylation go inside, dephosphorylation go outside. And these are two steps were different. So another part is using the circular permutant FP, the CPFP. So the original N and C terminals are fusing with a peptide linker and generate a new N and C terminals. So these new N C terminals can link the two other proteins. For example, in these conditions, they use a substrate of this kinase. So after phosphorylate by this kinase, and these two parts can binding together and cause a fluorescent light to emission. So like it changed the location of the N terminal and the C terminal because it's a circular structures. So there's a new N terminal. Before it's like in the center of the whole sequence of the GFPs. But this change can cause like can link to other proteins, and other proteins like approach and bind together may cross this GFP on the whole structures. So another is the intermolecular fluorescence biosense, and like the donor and acceptor, and when with the signal and these two sensor domain ligand domain like approaching binding together, and these two fluorescent protein are close enough, and so the donor of fluorophores can transfer the energy to an acceptor for chloroform. So the whole structures were different. A whole structure emissions and the light intensity were different. So this efficiency is depending on two factors. One is the relative distance, another is the angles between the donor and acceptor of fluorophore homophones. So for threads, so let's generalize backbone that this linker need to be very low, like 100 amino acid. So 100 amino acid will be how long? Okay, we can see that the intact whole channelrhodopsin will only like 300 amino acid. So this linker is pretty long. So this pretty long linker can cause that more flexible of these two N terminal and two C terminal, and the CFP and the YFP, their structures are quite similar, and it need a weakly sticky pairs. So usually it's CFP and the YFP. Cause these two approach together, and the conformation change type, and will cause the threat happens. Because of a high homology between YFP and CFP genes, which are the major IP pair used in intracellular threat biosensors. So of course, rather than the fluorescent proteins, they are either by or sensors that are based on cryptochrome with this cryptochrome 2 CRY2 with the LOV domain, that light oxygen voltage sensing domains. So it is binding this the flavin mononucleotide FAN or flavin adenodinucleotide FAD. So this is the kind of like a ligand. After with blue light, then the conformation change will cause the binding and the signal transduction activation of the CIB and the downstream signal pathway. So we already discussed about this, that most used is light oxygen voltage domains. And it is already reported that you can use it in the gene expression, gene editing, protein interaction, protein degradation, and second message. And this discussed in chapter 11 in details. So, so here recently there's also a redshift flash biosensors that is being produced. Is that this wavelength this response wavelength is approached to into the infrared, and this shows a previous those traditional flat pairs, and we see that the overlapped areas and it's here, but this overlapped area of emission excitation is better than this one, and also the light wavelength is longer, so it means that can be observed in a deep tissue. And also used in is the phytochrome B. These are coming derived from higher prompts, and because it has responded to longer wavelengths. However, okay, I will talk about that later. Okay, so when we absorb the red light and then combine into the PIF, that's the phytochrome interaction factor, binding to PAF, then it can be turned on by red light and turned off by far right light. It kind of a bistable activities. And FAB have a unique feature that both binding dissociation are controlled by light. So unlike the LV domain and Quite 2, the application of the 5B is being limited because the substrate that's the cannot produce a PCB by itself and need to purify the PCB and add it to the culture media. So that makes it impossible for this system using higher organisms like mice. To overcome these issues, some groups have developed the synthesized system and introduced a few genes of cyan bacteria, and there are 4 genes that are required to introduce these systems. Okay, to summary that the substrate the chromophore using is based on Hemi, so all this biliverdin, bilirubins involved in different binding with different chromosomes. So very recently, there's a nano body, and it's also called optobody, and it's quite similar to at the previous lecture. We're talking about the magnet. These are 2 different parts that cannot bind from on itself, but it needs an N and a P, like a negative and positive, and 2 different forms of ether dimerides. So it's also like the magnet that you have a P and have an N, so these 2 are binding together and then cause to have an intact chromosome, and it's called a nanobody. So there's in last year there's an update of this optobody that they use an LV domain, including nanobodies, so it can bind into the target body, and that's an M-cherry fluorescent protein. So with the blue light stimulations, and these 2 will dissociate, and this with the blue light emissions will go to the cytosol. And this is based on LV domains. Okay, that's all for today. So today's quote is all of the genetics, a relative new technology. Its potential is virtually unlimited. Okay, so today is for in May, so I continue to present a different chapter of the genetics, and enjoy a good weekend.